first, we're going to start with the respiratory system. And I have a few questions listed here. But before we get to those questions, I just want to outline a few things with regards to the material. And I think this portion, in terms of oxyhemoglobin dissociation curve that I want to talk about, will be extremely important, not only for this class, but going forward as providers, especially when you're doing any type of respiratory procedures. Certainly, it's going to be critical to know this when you're doing the procedure of intubation. So first, let me outline some things here. So I'm going to say, we're going to pretend this is your circulatory system. I'm going to basically draw the circulatory system. This is going to be the lungs that are going to be here. And at the bottom, I'm going to draw some cells. This could be any tissue cells that are metabolically active. So it could be liver, kidney, muscle, any type of cell that basically is doing work and essentially making carbon dioxide, right? And is utilizing oxygen to perform its functions. So we know that when we're breathing in air, right? Air, as it comes in from the atmosphere, has about 100 millimeters of mercury of PO2 or oxygen, and in the air it's about, let me use a different color, use purple, about 40 millimeters of mercury, right, of PCO2. That's basically carbon dioxide. And your red blood cells, right, or another name is erythrocytes that are flowing by here. They are predominantly composed of hemoglobin. So the red blood cells are basically, if you want to think about them, they're basically bags of hemoglobin. Right? And hemoglobin is essentially the protein that carries oxygen, and it also carries carbon dioxide. Right? It's composed of two subunits, 2 alpha and 2 beta. Right? We have them basically looking like that. It's a 4 monomers put together. So it's a tetramer. Tetramer is 4 monomers put together, right? And when it gets saturated with oxygen, basically every single monomer is going to be loaded with oxygen for a total of 4 oxygen molecules, right, that it could carry. So as this red blood cell, right, which is essentially a bag of hemoglobin, is passing in your circulatory system, right? The hemoglobin that's found inside your red blood cells will have those 4 subunits, 2 alphas and 2 betas. That's adult hemoglobin. And as it works in your circulatory system, it's going to be essentially the primary mover of oxygen transport, primary mover for your other, right, gases such as carbon dioxide. So the way this is going to work is as follows. So in the cells here, right, that I drawn, the difference in oxygen and CO2 concentration is different than as opposed to in your lungs. So in your cells, the PaO2 is about 40 millimeters of mercury, and PCO2 is about 45. So as the red blood cell, right, as it flows into your lungs, what it's going to do is going to pick up oxygen and it's going to offload CO2 here. And as it makes its way down, right, as it's loaded, now once it passes through the lungs, it has 95 to 100, right, PO2. And as it passes through the lungs, it has 40, right, PCO2. This red blood cell is going to be making its way here. And when it arrives, right, to the cells or to your distal circulation, we said it's about 95 to 100 PO2 and 40 PCO2, right, carbon dioxide. So, the flow is always from low pressure, right? Sorry, from high pressure to low pressure. So from high pressure to low pressure. So here, as we coming in, right, we said the oxygen content, PO2, was 95 to 100. So here in the cells, we see that it's 40. So the exchange is going to go from this red blood cell to your tissues. At the same time, the PCO2 is 40 millimeters of mercury, right? So the exchange was going to go from high to low pressure, high pressure being in your cells, right? It could be any cells, liver, kidney, muscle, right? Cardiac. And the CO2 is going to come here. And at this point, right, as the red blood cell travels, the composition now is different, right? After the exchange on the cellular tissues. So now as it picked up, right, the PO2 is now 40 and PCO2 is 45 as it made its way through the tissues. And then it's going to go back to the lungs and the exchange takes place again. Again, as I said, from high to low pressure, right? So as this red blood cell is coming to the lungs, PO2 is 40 and PCO2 is 45. And you see, right, in your lungs, oxygen is 100, PCO2 is 40. So from 100, it's going to come inside the cell to load oxygen. And this carbon dioxide is going to be essentially expired, right, as CO2, right, to come out from the red blood cell. Right? There's also a very important formula that I talked about in the lecture, right? And it's essentially, let me use a different color. It's essentially carbon dioxide. When it fuses with water, it makes carbonic acid, which further dissociates into a proton plus bicarbonate, right? So what all this means, it means that carbon dioxide is going to fuse with water. And water we have in our body, right? We're 60% of water. Then, as this reaction goes forward, is going to make carbonic acid, which is this thing here. And carbonic acid further dissociates into protons or hydrogen ions, right? And your bicarbonate. This reaction can go either way, right? Depending on the metabolic needs, right? So, for example, if I'm in my body, right? If I am having a lot of acid, right, or a lot of buildup of CO2, right, as my CO2 increases, an example I can give you is, let's say, somebody who is under the influence of heroin and they're not breathing adequately, they're not excreting their CO2, they're retaining CO2. This reaction, right, is going to go this way. So CO2 that's being retained in their body is going to come together with water. It's going to make more carbonic acid, and we're going to have more protons in the body, and the person is going to become more acidotic, right? The same way, if you come in your system, you start to ventilate them with a BVM, and perhaps you are doing it at a more rapid pace than you should be, right? You're going to move the reaction to the other side towards the reactants. So you basically, what you're going to be doing is you're going to be blowing off CO2 as you ventilate them. And what you're going to do is you're going to be combining bicarbonate with hydrogen ions. The carbonic acid that's in their body is going to be basically be broken down into water and CO2, and this CO2 will be excreted out, right, through the respiratory system. So this reaction works either way to maintain acid-base balance. This reaction works in your respiratory system. Later on, when we learn about kidneys, we learn about the renal component, right, the bicarbonate buffer system there. The respiratory component takes minutes, 2 hours. It's relatively fast. The bicarbonate system is a little bit longer. With the kidneys, it'll probably take days to weeks to start working. So in a nutshell, right, this is how hemoglobin works, how it offloads right, oxygen, how it picks up carbon dioxide on non-iron sites, and this is the transport right in your system. So the next thing that I want to show you is basically, this is how the hemoglobin molecule looks. We have iron in the center, and it's bound to the nitrogens here. These rings called porphyrin rings, and together with iron, they make heme, right? So all this is called heme. And you notice the oxygen is usually it's diatomic molecule O2. That's how it exists in the atmosphere, and it binds here, right? Let me ask you a question. What else does iron bind in this specific location where it binds oxygen? What else can it bind here? Does anyone know?
CO.
Carbon monoxide.
Yeah, carbon monoxide. So the other thing it can bind carbon monoxide, right? This is how the molecule looks, right? This is here is carbon monoxide, and this has affinity of about 200 times greater than oxygen, right? So carbon monoxide has 200 times greater affinity to iron. Now, what I want to stress to you guys, which is very important, this iron here, it does not, I repeat, does not bind CO2, hydrogen ion, or anything like that. Those things are bound, right? Either on carboamino, right, terminals like here, right, or on the other amino acid residues on the hemoglobin molecule itself. But this portion here, the iron portion in the center here, the only predominant things it binds O2, right, which we want normally, or in cases where a patient or person inhales carbon monoxide bind here. There's other things it could bind, such as nitric oxide, but we're going to talk about that later. I will probably talk about that once we get to cardiology chapter. But for now, what you need to remember, the preferred is O2. And if someone happens to inhale CO, carbon monoxide, it will bind there. And when carbon monoxide binds the site, it binds so strongly that it switches the hemoglobin molecule into the relaxed state. So I'm going to show you, there's two states. There's tense state, and there is relaxed state. And in the relaxed state, the affinity is so strong that even if there was other O2 molecules bound to hemoglobin, right? Let's say if we had 4 of these, because hemoglobin is 4 of these monomers put together. So 1, 2, 3, 4. So if, let's say, one bound carbon monoxide and the other 3 had oxygen, because carbon monoxide binds so tightly, the other oxygen does not want to offload when it gets to the tissues because of this strong affinity to it. And it switches the hemoglobin into this relaxed state where oxygen is bound, but it doesn't want to release it. And that's what we see when we have CO poisoning, carbon monoxide poisoning. Even though there's some oxygen that may be bound on the hemoglobin, when it reaches the tissues, it doesn't want to offload. So basically, tissues are oxygen starved, right? So this is your structure. This is what I was basically diagramming out, right, for your red blood cells, right? So this was your red blood cells. Another name is erythrocytes. If you recall, let me ask you this, from your bone and skin chapter, where are erythrocytes produced?
Bone marrow.
Bone marrow.
Bone marrow, excellent, right? So we have the hematopoietic stem cells. They differentiate, they basically subdivide, and then the myeloid line, right? They subdivide into platelets, red blood cells, and eosinophils, basophils, and a few other cells. But basically, what you got to remember, the stem cells, right, in your bone marrow are the ones responsible for production of red blood cells. So once red blood cells are produced, they mature. Usually, the lifespan of red blood cells is about 120 days. So after 120 days, they die. What you want to remember, and it's very important for red blood cells do not have nucleus, and they do not have mitochondria. So if they don't have nucleus and they do not have mitochondria, it means because they don't have mitochondria, the only thing they can do is glycolysis for their energy needs. They cannot do oxidative phosphorylation, right, in the mitochondria because they're lacking that organelle in it. So here, as I was explaining, right, this was the formula, right? As you saw, CO2 comes with water, makes carbonic acid, and then dissociates into hydrogen ion and bicarbonate. In your alveolar space, right, in your lungs, the oxygen content is about 100 millimeters of mercury. Carbon dioxide content is about 40 millimeters mercury. And here we have the red blood cell. Oxygen gets loaded in the lungs, right, onto the hemoglobin molecule, specifically at the iron, right, Fe2+, which is the ferrous form. So oxygen gets loaded, and here we see, right, the carbon dioxide that was there from the tissues, it gets offloaded into the atmosphere. So then this red blood cell travels to the tissues. In the tissues, we see that oxygen is low because tissues utilize oxygen for their metabolic needs, right? And we see oxygen is 40, and carbon dioxide in the tissues is 45. So as I said, the flow is from high to low pressure, right? So high to low pressure, red blood cells is loaded with oxygen, so oxygen is going to go into the cells. And here in the cells, the carbon dioxide is about 40, the CO2. So that's why from the tissues, the CO2 is made. Why the CO2 is made? Because the cells are utilizing it, right, as it's a byproduct of metabolic metabolism. So as the cells work and perform their functions, right, CO2, hydrogen ions, right, lactic acid, right, as let's say you're working out in the gym, right, you have byproducts, and all of them are produced, and they travel back into, right, the erythrocytes pick them up, and they transfer them back to the lungs, right, as this makes its way back to the pulmonary circulation. So hopefully this makes sense. All these numbers, I know it looks like a lot, but if you remember, the exchange is from high to low pressure, and the major gases that are exchanged is oxygen and carbon dioxide. Is that clear, or anyone have any questions?
Pretty clear.
Okay.
So clear.
Now, if this is clear, we're gonna go and I want to explain. We're gonna get to the oxyhemoglobin dissociation curves. This is what I was saying to you guys about the tense state and the relaxed state. And let me kind of make it, maybe you will kind of to start to understand this better. When the hemoglobin is in this tense state, right, it wants to give off oxygen. So it wants to get rid of oxygen that it has in all these subunits. So let's say if this was alpha subunit 1, alpha subunit 2 here, right? Beta subunit 1, beta subunit 2, right? So 4 subunits. In the tense state, right? Here we have different charges on the amino acids. So we have pluses, minuses, and they make connections, right, with each other. And in this case, when it's in a tense state, hemoglobin is desaturated and oxygen does not want to bind. And also in the same sense, it wants to get rid of its oxygen. So it wants to give off. The tense state is usually you see is happening in the tissues, right? Tissues. Why does it occur in the tissues? Because we said in the tissues, we have buildup of CO2, we have buildup of hydrogen, right? Acidosis, lactic acid. And all those factors, they will essentially stabilize the tense state, right? They will stabilize these charges in the tense state, and oxygen will offload, and oxygen will not want to bind. On the other hand, in your lungs, right? Lungs, where oxygen PO2 is about 100 millimeters of mercury. As one oxygen binds to the heme group, right? There is something known as cooperativity, right? And means they cooperate together. As one O2 binds, it makes the second one bind easier, and the second one allows the third one to bind easier, and then the fourth one binds very easy. So in the lungs, it switches conformation, right? And this happens with specifically the histidine amino acids. I'll show you another diagram how it works. What they do is they pull, right? When the oxygen comes, binds, the histidine pulls on the molecules itself. It changes shape from tense state to the relaxed state. And the thing about the relaxed state, yeah, it loads oxygen, but in the relaxed state, it doesn't want to give off oxygen so easily, right? And it makes sense because in the lungs, I want it to load. I want it to load up with oxygen. I don't want the oxygen to dissociate. So in the lungs, it's usually in the relaxed state, but in the tissues, it wants to be in the tense state. But remember, we just talked about carbon monoxide. What carbon monoxide tends to do, it kind of, when carbon monoxide, let's say, let me just make one molecule carbon monoxide here. If carbon monoxide, right, binds on one side, what it does is switches the conformation to this relaxed state. And even though when this gets to the tissues, it will maintain this R state, relaxed state, right? And even though it's loaded with oxygen, it will not want to give off, right? It's basically, think of it, it's car occupied with oxygen and the doors don't open. The doors are closed and it makes its way through the tissues. Nobody wants to come out. The reason why is that the relaxed state has high affinity. You know, another way of saying it, it has high affinity, or it has high bonding to those oxygen in the presence of carbon monoxide, and it doesn't want to give this off. On the other hand, right, if everything is fine, the oxygen is only loaded, there's no carbon monoxide. These things, the byproducts, the CO2, hydrogen ion, lactic acid, right, carbonic acid, they will basically come in, bind on residues other than the iron. They will change the relaxed state into the tense state, and oxygen will dissociate, right? So hopefully that makes sense. And this is the how it basically functions, how it looks, right? So this is your iron, and this is your oxygen. So the moment iron binds to this oxygen, it kind of pulls from this blue state to this red state. So you see how it became more like linear, right? Linear. And this change in shape of the molecule is what switches it from the tense state to the relaxed state. And this usually occurs in the lungs, right? Where two binds. And I want this because I want this loading to occur. So they will say, right, as iron 2+, which is the ferrous iron, it moves downward to meet with oxygen. It basically is going to pull the histidine residues here, and is going to cause the switch from the tense state that gives off oxygen to the R state, relaxed state, where it picks up oxygen. Right. So this is what I was basically explaining. The other thing that I want to point your attention to is, you know, why is blood red in color? The reason why it's red in color is that once iron binds with oxygen, it essentially, that what gives its color. When it's not bound, right, it's without oxygen, we'll have the purple color, which you see in venous blood, right? The dark colored blood, deoxygenated blood, and this is why this is. So O2 binds with iron, essentially gives it its red color. We talked about the relaxed state and the tense state. Any questions about that? About tense to relaxed state? This will become important as we talk about hemoglobin dissociation curves. Or that's pretty clear.
So tense state is the left shift?
The tense state will be the left shift, is correct. So sorry, okay. Right. Sorry, the tense state will be the right shift, is right
shift. Okay.
Tense state is the right shift. It wants to get rid of oxygen. The relaxed state will be the left shift, where it holds onto the oxygen. So I'm just going to repeat it one more time. Tense state, right shift. Relaxed state is left shift.
And the tense state, it's caused by its environment, like it's because
yes, that is correct. So usually there was a scientist named Bohr, right? He was they call it the Bohr effect. So the more CO2, right? So let me go back to this thing. So the more CO2, the more hydrogen ions, the more acid you are making, these will bind onto allosteric sites, meaning not the actual iron sites. They could bind to residues on the hemoglobin molecule. And what they will do is they will stabilize these charges, right? They will cross-link them. They'll stabilize these plus-minus charges on the amino acids, and it will switch the relaxed state into the tense state in the tissues. And this occurs in the tissues. Why? Because tissues are metabolically active. They're making carbon dioxide, they're making hydrogen ion, they're making lactic acid, right? Carbonic acid. And all this acidosis, right? All these factors will basically switch from relaxed to tense state. In the tense state will cause a right shift on oxyhemoglobin dissociation curve. And the tense state wants to give off O2, right? You see how it says low O2? Because at this state, it doesn't want to bind. It wants to get rid of it. And all this, it's called the Bohr effect. Right? Maybe you heard in chemistry, there was Neil Bohr. This guy, Christian Bohr, was his father. So it's after him. Bohr effect means all these things, carbon dioxide, hydrogen ion binding to hemoglobin, switching it to the 10th state. Does that answer your question?
Yes, thanks.
All right. So we talked about that. This is, I explained this, but this is just gives you a numerical or graphical data, right? A stands for arterial blood, V stands for venous blood. AV difference means arterial minus venous, and this is what you get. So we said in the PO2 in the arteries, right, is 100, especially when it gets to the lungs. And on the venous side, oxygen is 40. Why? Because they utilize oxygen. The difference is 60. Here, they just give you saturation. So they say saturation of hemoglobin is 97.5, or usually reaches 100% in your arterial side at the lung level. And the tissue is right at 75%. Why? Because about 22 here, they say, but maybe 25% gets offloaded and gets utilized. Another important thing to keep in mind, they show you here in the venous side, the O2 bind to hemoglobin is 15 milligrams per deciliter. That's what, if you go to your doctor, they'll check your blood work. Or if you're in the ER, right, they will check what's the H&H. They'll ask H&H. That stands for hemoglobin and hematocrit. The normal level of hemoglobin is 15 milligrams per deciliter. Hematocrit is about 45%. So that's the measure of how much hemoglobin is in, saturated at that point. Why I bring this up, right? You notice that even though it's in the venous side, it's only 75. So tissues extract 25% from it. Why? Because as I said in the respiratory lecture, right? We breathe episodically, means I take a breath in, I exhale, I relax, then I take a breath in again. But in the tissues, the exchange has to be continuous. So that means if I, didn't breathe for a little bit, right? The venous blood still has 75. So the next cycle, let's say it circulates in the lungs, but I wasn't breathing at that point. There's still oxygen loaded on it. So when it gets to the tissues, it could still extract oxygen through it. So it's the example you may see is, right, the guy who is not breathing well, maybe secondary to opioid overdose. He's breathing 6 times, 4 times a minute, right? He still can sustain some, oxygen delivery, because even though he's not getting 100% saturation, that 75%, that goes the second cycle, the tissue can extract. But there's only a time where, this can go on. If this prolongs for, too long, right, he will go into respiratory failure and respiratory arrest. So this is just the differences here. In the patient who is, for example, septic patient, their venous consumption becomes much higher. So in the hospital, we may measure their venous oxygen content, right? How much saturation is on their venous side. So we know it's about 75%. But let's say we check that septic patient's level, and instead of being 75%, it's 55. That means that the tissues is extracting 45% every time it goes there. That means that tissues level is much higher. They're metabolically active, right? And in sepsis, you have fever, you have high metabolic demand, the heart rate is elevated, right? And it makes sense. Tissues require more oxygen to perform their function. So they will pull up more oxygen off that. So, this is just to give you an idea, right, when we measure these things. The other thing I want to talk about is cyanosis. What is cyanosis? It's I showed the picture, basically hands had this bluish coloring. You may see circled oral cyanosis in kids, right, if they're not getting adequate oxygen. So what a cyanosis basically means, the amount of hemoglobin that's desaturated. right? So they make this important distinction. It's not from absence of saturated hemoglobin. It's essentially from the presence of desaturated hemoglobin. And why? Because like they give you an example, you may have an anemic patient, anemia, right? They may not have enough red blood cells in the circulation, but all those red blood cells are oxygenated, meaning, let's say instead of having 5 to 6 red blood cells that are circulating, let's just say they have half of that. Let's say they have 2 to 3, but 100% of them are saturated. And so even though they're cut in half, right, you will still see, pinkish color, right? You may not see cyanosis. Why? Because they are fully saturated. So when you have cyanosis, it means that you definitely have presence of desaturated hemoglobin. So that's very worrisome. That's why we provide oxygen. So just to aware you of what cyanosis is, right? We talked about this, right, in terms of what makes a right shift. The right shift is elevated carbon dioxide, hydrogen. Temperature will also do this. Where does this occur? This occurs with metabolically active tissues. I just gave you an example of a patient who has sepsis, right? They'll have a fever. The tissues utilizing all the oxygen they can. They produce CO2. They produce hydrogen ions, right? So they will be metabolically active. What's going to happen to hemoglobin? Hemoglobin is going to dump oxygen, right, in that setting as it gets to the tissues.